Okay, so after that insightful talk by Dr. Damle, I'm sure you all are very energized and very excited to put your brain into good exercise, right? So here I would like to give you some insight about the way we can go ahead doing research and some ideas to kickstart. So, and I also would like to urge that this particular platform, take this as an opportunity or a stepping stone to transform your thinking in the direction of vigorous and innovative thinking, okay, in scientific works, in the future also. So first of all, what's this research all about? Research must be the most cliched word you all would have heard, right? Everything has research, researched cloth, researched textile, or any researched juice. But what is this actually research? Research is basically a mindset, actually. So I would say I always like to give these two examples when I talk about research. The first one, this is for the reason why we need tools to verify our observation. Of course, observation can be very beautiful, but is it good to put it to acid test? Okay, so this one simple example proves that it is important. You must have seen these illusion. Actually, it's an illusion. Even if I say it's an illusion, at the end of the day, even if you believe that it is not true, in the mind you want to believe that both these lines are of same length, right? So all we need actually is a measuring tool or the scale to tell you that this is an illusion and both of those lines are of the same length. So that's what scientific tools do actually. Next one, you must have seen this. This was actually done, I think for a long time till Galileo did this experiment. Many would think that balls of heavy weights, the one mass, the object of heavy weight would reach the ground faster. But when it was shown that both would reach, of course, in vacuum without being affected by air pressure or air, the breeze, they would both reach the ground at the same time. But for this, actually, all you had to do was carry out that experiment. Just hold two objects of different masses and just drop yourself and see. You can see which one falls faster. So this is what you need, how the thinking should go. That's what is basically research is all about. It's not just holding test tubes or, working in lab with the lab coat. It's basically the thinking or the mindset. So the science itself, I would say, it's not the knowledge. It is basically the best tools that you have, which actually can be put to whatever that you have observed or objective, the subjective observations to quantify and to objectify it. So these should be used as tools to test all our hypotheses or whatever we think. And once you start doing that, in every aspect or any hypothesis you come across, you will start analyzing in this critical fashion. So it's basically a methodology of thinking and working. And very importantly, the three qualities that it can have is demonstrability. That means you can show that it is, yes, when I drop the balls, yes, they fall in the same speed. Then reproducibility, which is very important. Whether the other person who repeats your experiment or who follows your hypothesis also has the same result. Then comes a very important is fallibility. In any critical thinking or in any kind of hypothesis that you put and enumerate, you should think whether I can be fallible. Yes, fallibility is inherent quality of everyone, including the science. That's why it sharpens its tools continuously. Many a times we feel that science is stupid that today it said something, tomorrow it says something. No, it's actually sharpening its tools and making the observation, understanding it critically and giving you better result every day. Because the bias can easily enter in our observation. When we see how you saw the Muller layer illusion, however we dismiss, inherent bias can always come in. That's when we say we need to design the protocol. That's what is all this exercise about. How you can do the research protocol design devoid of bias, how we can avoid the bias, possible bias. I'm sure more details about this. That's why the statistics is all about actually, which will be given by Dr. Shana and Dr. Pratibha will talk more on this. I just want to tell few types of very important bias that can come is information bias. When you start collecting data which is inaccurate, or it could be selection bias. You're not randomized the selection at all, or you have selected wrong category of patients where you are not supposed to give any medicine. For example, I did see one of the study where they had included pregnant women in the study. So what happened? In fact, pregnant women should not be included in the first setting because ethically it is not allowed. Such kind of selection, lack of randomization or wrong selection can make your design poor. Third one is very important, confirmation bias. Confirmation bias is, I think, very common. When we like something, we'll try to gather evidence in a way that it supports our view. For example, if you like coffee, you will get all the data or evidence that says that coffee is very good for aging. Okay, that's one thing. So this is what we tend to do when we design the protocols. So these are the bias which we need to avoid. So how can we avoid? Of course, there's no first thing and foremost thing would be we should be self-aware and honest about what we think and how we can design, where we can go wrong. We can plan that well ahead of study. And well ahead, planning well ahead always gets you good outcome. That's very important. So take more time in planning so that you don't falter at later stage. So have a good clarity on objectives, clarity on the outcomes, how well you can blind and remove the biases, also address those earlier three sources of bias that I said. As I told, this will be well explained during later sessions by Dr. Shana and Dr. Pratibha. So in this particular research protocol competition, we are concentrating on 4 ideas. Of course, you can come out with more ideas. These are just pointers. Research is a vast topic and sky is the limit. You can always come out with better ideas, and that's all about, it's all about research actually. So we have, to make it things little more easier, we have suggested 4 kinds of arms to this research protocol, which will either you can take up on Atrimed products. Okay, Atrimed products also, in fact, we have made it a little easier, have given you some suggestions of the products, but you can take up any product and you can work on that. Or you can think of some basic science principles like Siddhantas and come out with some design a study on that. Or you could go in for an Ekamulika Prayoga or a single drug medication. Or apart from all this, you can come out with any novel ideas you think can be useful. And in all these 4 groups, you can choose any in silico, in vitro, in vivo, or clinical trials, depending on the feasibility. You must also, because while designing, you should also think about the feasibility of the portion also, not just selection. Okay, so you can plan accordingly. So in the first segment where we have Atrimed products, we have suggested 7 products. Of course, these will be recorded so that you can go back to this and look later as well. Either you can think about a combination drug of Viscovaz with the Zeptico. These are basically for wound healing and secondary ulcers. Then could be Spasix, which can actually work as a single drug remedy for asthmatic attacks. So you can choose either it is an acute case or the chronic case. Of course, you also have to plan the duration of the study, because I'm sure in housemanship you have the constraint of time. So think about and what best parameters you can use. For example, whether you will use pulmonary function test before and after. It could be just 2 months study or 3 months, whether there is any improvement in the pulmonary function. So another could be a clear, which is again, the reason why we have also thought about this is these can be quickly done. For example, acne doesn't require many number of days to get cured. Similarly, you can have kinase, which is for pain management. You can have pain scores and manage design accordingly. Or it could be an upper respiratory infection or acute rhinitis, flu-like syndrome, for where you can use Viridol, and it could be just 5 to 7 days of treatment. And or you could also think of anti-dandruff remedy, Scurfol. Try that. Or you can think of any type of psoriasis, where you can use Atrisor ointment and Atrisor capsule. More details of its dose and other things, you can definitely check the product details, or you can come back to us as well. Some suggestions in basic science. These are, as I am saying again, these are just pointers. You can come out with your own ideas. You can take more help from your mentors, tutors, because we have best of the brains out there. So you can get many informations and you can come out, design a very good study, I'm sure. So just to put some pointers, I have given some examples here. You can think of role of Viruddha Ahara. But what I would suggest is, since the time period is short, I would suggest to be specific about what you want to expect, or what the primary objective or outcome should be. So role of Viruddha Ahara in immune response, or in the inflammatory response of, whether it has any impact. You can study on that. Or is there any relation between the way we respond to inflammation in our prakritis? Or is there any relation between rasa guna virya vipaka that correlates to class of phytochemicals? For example, if you have madhura gana, or these kind of virya guna virya vipaka, do they all come in terpenoids? Do they all come in, say, if you have some bitters, tikta rasa dravyas, do they all come in alkaloids? So these kind of class of phytochemicals you can assess. Of course, not just literature review, you can test it using HPLC, HPTLC, and such other instrumentation. Similarly, you can also, you must have seen, we have 20 fresh plants. The Ayurveda suggests them to be used as fresh drug. So you can really see, by using them in fresh form or in the different extracts, is there any difference in the phytoconstituents actually? Or is there any single drug versus gana? When there is if you see, there are groups of single drugs. For example, Vayastapana gana or Sanyajanana gana. So if you take single drugs, each of them, do they have any similarities in the LCMSP? So do they have any differences? What is it? That you can study. Similarly, can Samaprakriti person have a differential immune response? Does he have a better immune system? Of course, this needs good amount of blinding, with many people assessing the Prakriti, so that we don't need to, we don't have contradictory assessment. Similarly, you can also think whether the absorption of drugs on Pushya Nakshatra will be better compared to any other days. I'm just giving Pushya Nakshatra as one day. There could be many. If you see Kaumara Vritya or any other Samhitas, you will get, this has to be given on this particular time. It is well mentioned. So you can study those and come out with ideas. Upavasa is another, Langana is another very important concept that you can study on. You can see whether there is any inflammatory or allergic response with the blood parameters like ESR, CRP, IgE or IgG responses. Also, you can think of Shodhana effect or effect of Vasti on gut microbiome. We can do the microbiome of the fishes, genomic analysis and find out that. So these were just the pointers. As I said, you can come out with much better ideas. Similarly, next set of studies could be Ekamolika Prayoga, that is single drug usage. So this, you can have ideas from Agre Dravyasthana, where single drug is said for most of the time. For example, Rohitaka is said for liver ailments, but many of the medicines that we use in actually liver complaints does not contain Rohitaka. Of course, there are some, I'm not saying there are no, but it's hardly, we have not seen this kind of single drug being used in that particular where they have said Agria. So you can really see whether different dose forms of the same drug can have differential results, or same drug in different types of those, jwara. When they say jwara, is it all kinds of jwara, or are there any typical pattern to it? You can study those things, or you can get some idea from, as I said, ganas also, like vayastapana, kandugna dravyas. Are they specifically, why have they been put in one particular gana? Are they all have similar effects, or they have any different patterns of different kind of disease with similar symptoms? That you can study. Similarly, you might have seen one haritaki has been told either, either as a powder, it has different usefulness. When it is boiled or steamed, it has different efficacy. Actually, does it have any differences of phytochemistry also when it changes? We can check that. Whether the efficacy difference also occurs, of course. Really, the one is used in Atisara, one is used in Gulma. So you can check whether there is a difference in efficacy as well. Not to forget Anupana, because with the different Anupanas, the effect seem to be different in as per text. That also you can put to test to see that is possible. Well, doing this, planning this is one thing. Designing to have a proper outcome is also another thing. Last, very important, is novel ideas. Of course, I don't need to put any guidelines or I don't need to give you any pointers, because then that never becomes a novel, right? But still, it is, I do have some novel ideas. If somebody comes out with that, I'll be very happy. So just give you some pointers which you can think of. Can you make self-dissolving bandage for non-healing ulcers? Say you put the bandage. We do have, in fact, biodegradable bandages available now, made of silk actually. So can we incorporate medicine in that and make a self-dissolving bandage for non-healing ulcers? Can we see whether we do use Kshara Sutra, right? Can we develop something, a liquid that solidifies as it goes, traverses through the Bhagandhara fissure tract, and probably is more easier to apply? Is it possible to have some intranasal medication to treat CNS disorders, or even some systemic disorders through intranasal? Because we have whole host of medications suggested in Nasya. And of course, some very novel thinking where we don't have a good answer at this point of time. Can you really stop that hair follicle destruction? Because that happens not just part of aging, it's more to do with the hormones. Can we have something in the last phase where it destroys? Can we inhibit that particular enzyme so that hair follicle destruction can be stopped? Or can you have any other methods of tissue culture, or aeroponics or hydroponics and such other systems to have sustainable delivery of active compounds? Why I say this is because for every medicine we need to use a lot of plant sources, right? So we need to make at one point of time these medical compounds, the active compounds, sustainably available. So can we think of such? Of course, I'm sure you will come out with more ideas. So at the end, I want to tell that we are here to help you. So I just gave you some pointers. As I told earlier, we can do all these works, either take it as in silico work or take it as in vitro. You can plan your studies accordingly. Pilot studies, blind, randomized control studies, or anything. But at the end of the day, I want to assure you that we are here to help you and we want you to excel. And take this as an opportunity to have a innovative thinking in future also, so that we get much more fruitful ideas, and well, we can harness the beauty of Ayurveda. Of course, it's a competition. I'm sure we have made it competition only to make you more active. But as I said, work with a vision to continue forward the scientific thinking. Thank you very much. Wish you all very good luck. If you have any questions, you can ask me now, or you can write to us later, however the time permits. Yeah, I see a question here. Research is to be done. Final year students can definitely join. But one thing is, I'm not sure if interns are allowed to prescribe drug. I don't think so. You will have to have the supervision of your teachers and professors in your college. Pop in your questions in the chat box if you have any questions. I see one question. Will date of submission of protocol be extended? The current deadline is January 15th. I think we have enough time. You still have some more than a month to prepare. Because if you extend still more, if you don't submit at least by Jan 15th, I don't think your 3 months or at least 4 months of study can be finished. Yeah, everyone can see there's an answer there. Stage 1 submission starts from 15th December and the last date for submission is 15 January 23rd. You can choose from any of those 4 topics that I just mentioned. It will take some time for the next speaker to start his session. So probably till then you can think of questions if you have any, and we'll be available here to answer the questions. You can either open your, you can unmute yourself, or let me know if you want to switch on the video and ask. Yes, this is going to be recorded session. You can watch it later also. I will check with the people here how they can send you the links to view the recorded version. The link will be sent to you. There's a question here where we have to submit the protocol. So we will send you the link for that. I was talking about Dr. Dharmaraj's question. Yeah, it's a very good question. You are asking about modifications in medicine. That means you are asking whether we can have different dose forms like injection or IV fluids. Definitely you can think of dose forms. But only thing is you will not be able to ethically allow to test it anywhere. You will have to have lot of preclinical works before taking it forward. But you can definitely plan the study, the basic studies for that in this particular protocol. And for those who wanted to know where the submission has to be made, it is at www.atrimed.com under research. You can also study PK parameters. For example, when I said single drug or combinational treatment, you can see whether when you combine the drug, what is the bioavailability of one particular marker compound. You can test in patients. You can see whether they are in the serum, the bioavailability of one particular active marker is being supported or being increased with the addition of other drugs in the kashayam or group of medicines. You can try those studies. It can be done in vitro or in vivo also, actually. In the rat model studies you can do, or in healthy volunteers also, if the it is a very common drug that we use. Using just ginger alone or tricotoo, the combination, does it make it potentiate the benefit? That you can see whether any marker that you think is not very desirable, like a toxic compound, whether that goes down if you add the other drugs. You can study those kind of works. Similarly, these can be, you can plan in silico studies. I don't know how many of you have done an in silico study where we study the interaction of drug with the protein of interest or the target protein, and see its binding reactions, whether it really binds to that particular. For example, you can think of a disease and the well-established targets for that. For example, if you say alpha glucosidase enzyme in diabetes, if you want to study some anti-diabetic drugs which can have this particular activity, that we can try in silico first and get a clue, and then you can proceed for in vitro studies. But when you think of any study, as I told, first plan the study well. Discuss with your mentors, discuss with your tutors. You can also reach out to us for any help regarding designing of the study, or whether even ideating process, and then assessing the feasibility, whether we can really take it forward, such studies. Looks like there are no questions, or probably thinking process has started, I guess. Think where you can make difference. And when you choose this study, I would suggest use those critical thinking because recently I saw one study where they said, the sitting on the ground and eating is good because when you bend, it presses on the pancreas. It might press on the pancreas, but doesn't it press on the kidneys also then? Right? So I think you need to be critical when you claim. I'm not saying that this is not good, but is it really that good? Is another thing affects? You need to think critically. If there are any questions, do post in. We have some question, ma'am. The aspect of dosage of Anupana isn't mentioned in our Samhitas very precisely. Is there any scope on doing research regarding that dosage of Anupana? My understanding goes right, we do have different Anupanas. First, I think you can see whether those Anupana, the relevance of those Anupanas. Now, whether I use 1 spoon of honey or half a spoon of honey, it is discretional call. Most of the things, I think, sometimes we have to think logical also. When the Anupana is mentioned in Ayurveda, definitely our ancestors wouldn't ask you to take glass of honey, right? So it is that much amount that is needed to probably mix that particular drug. I think we should take that logical and first assess the basic relevance of that Anupana. Then maybe you can definitely modify the drug, whether you need only just 1 drop or 2 drops or 5 drops, you can definitely, right? I hope that answers your question, Gayatri.